Okay, this is a continuation, part 2 of the male reproductive system. So we're going to start with spermatogenesis. And spermatogenesis occurs within the seminiferous tubules of the testes. It's stimulated by, believe it or not, follicle-stimulating hormone, which we oftentimes think about as a female reproductive tract or a female hormone. But follicle-stimulating hormone initiates spermatozoa production. However, testosterone is also required for spermatogenesis. There are about 300 to 500 million sperm made per day from puberty to death. Spermatogonia, or stem cells that produce sperm, start producing sperm through meiosis, which we will talk about just really quickly. It'll form, the spermatogonia or the stem cells are formed during embryonic development, and they remain dormant. They don't do a whole lot. They just kind of remain quiescent during puberty. And so basically what ends up happening is the spermatogonia are divided into two cells. One remains a stem cell, and that's part of the definition of a stem cell. When the stem cell divides, it produces a second cell, but it also reproduces itself. And so what ends up happening is this cell here, the offspring, gets pushed towards the seminiferous tubules and becomes a primary spermatocyte, which we'll go through in just a few minutes. The spermatocytes then continue undergoing meiosis. So the stem cell itself regenerates itself, and then this starts dividing to produce 4 sperm. And again, we'll talk about that in just a minute. So if we look at a cross-section through a seminiferous tubule, so this is deep within the testes, you'll see different layers. So over here, here's some capillaries with vasculature. You'll see some connective tissue, basement membrane. Basically what we're looking at here are different layers or different levels of spermatogenesis. So we have the spermatogonium right here, the primary spermatocyte that's going to prepare for meiosis 1. So this cell is the one that just divided, gave rise to a second cell, and regenerated itself. So basically, this is the stem cell, okay? The sperm stem cell. This primary spermatocytes are going to start undergoing, meiosis. And again, we'll look at that in just a minute. And so as you see the cells undergoing meiosis, they're going to get closer and closer to the lumen so that they may be released to the epididymis. So how much of this do you need to know for the exam? You need to know where the primary spermatocytes are. The secondary spermatocytes you're going to be finding adjacent to the primary. These sustentacular cells are basically cells that are the product of meiosis, and then the spermatids are going to be, basically in this region, the spermatids are going to be developing from these sustentacular cells, all right? On the left-hand side of this, you're going to see, so here are our primary spermatocytes over here. The secondary spermatocytes, you are defined by being able to see the actual mitotic figures, and then eventually you'll see the spermatids completing spermatogenesis. However, these spermatids are still not completely developed. So it's a little easier sometimes to look at these pictures in terms of a cartoon. So the primary spermatocyte is going to be, again, the cell that's going to be undergoing meiosis. The spermatogonium, this is the stem cell, okay? Why do I keep talking about stem cells? Well, it's important to understand that this cell is going to be producing something that then can enter meiosis, but it's also, and it's showing another stem cell being produced. I oftentimes draw it like this. As the division occurs, you get 2 cells. The product is going to be one primary spermatocyte. The other one is going to be a spermatogonium. The primary spermatocyte then undergoes S phase, which then will complete meiosis 1 to produce secondary spermatocytes. After the second division, you end up with spermatids. These are haploid, and actually you end up, remember at the end of meiosis 1 here, this is where you have the reduction division happening. The secondary spermatocytes are also haploid. Right here, the primary spermatocyte is diploid. If you need to go through and review meiosis, please feel free to do so. After meiosis 2, or that secondary division, the spermatids then are going to undergo secondary physical maturation, where they can become haploid spermatozoa, and that happens in the epididymis. So primary spermatocyte divides into two to make secondaries. The secondaries divide to make spermatids, and then the spermatids undergo maturation. So each spermatid then turns into a single spermatozoan. Spermatozoan is singular, by spermatogenesis. And so the spermatids will be embedded in the Sertoli cells, or the sesenticular cells, and then they will gradually develop the appearance of mature spermatozoa, and we'll see a picture of how that happens in a minute. Spermatization loosens the attachment, allowing the sperm to enter the lumen of the seminiferous tubule. And this whole process takes about 9 weeks, so it takes a while to make a mature spermatozoan. So during week 1, we start with the spermatid. So this was at the end of all those meiotic divisions. Over time, you'll see that this one region called the acrosomal vesicle starts to grow. Over time, you'll see a reduction in the number of cellular components, intracellular components like the Golgi and so forth. The mitochondria begin to aggregate around an extension that's going to become the flagella. And by week 5, the sperm is divided into 3 segments: the head, the neck, which attaches to the middle piece. Right? The neck is kind of a part of the middle piece here. And then the tail, which is a long fibrous flagellum that has an array of microtubules to allow the flagella to swim to contact the ovum. So the head contains the nucleus, right? And that's going to contain the DNA. The middle piece has the mitochondria that are going to be really, really important for producing energy for the tail to whip back and forth for swimming. And then the tail, like I said, is a very long flagellum. So the mitochondria, as it turns out, as fertilization occurs, only the head enters into the nucleus of the ovum. The tail and the mitochondria remain outside. Therefore, only every zygote only gets mitochondria from the ovum. So all of our mitochondria came from our mom because, for the most part, like 99.9% of the time, the mitochondria from the spermatozoa get left behind after reproduction. So moving on to the epididymis, where the sperm mature. This is where sperm are going to be producing that really long tail. So basically, the epididymis can also be called, I guess they call it sperm boot camp. The sperm will be released in the seminiferous tubule. At that point, they're not functional. They're not mobile. They cannot swim. There's no possibility of fertilization. The epididymis is on the posterior border of the testes, and it's superior to the seminiferous tubules. The epididymis itself is a really long coiled region. There's the head of the epididymis where the sperm is received. The body, which is distal to the ducts of the mediastinum. I should have said the head receives the sperm from the mediastinum. Forgot to mention that. And the tail is the region where sperm is stored. And so I'll show you a picture of that in a minute. And so sperm leaving the epididymis, it turns out, still are not mobile. The sperm need to be activated. So there is a peristalsis of smooth muscle that allows the transport of sperm from the epididymis out into the ejaculatory duct. And so this is what we're looking at here. So here is the ductus deferens coming in, wrapping around the posterior side. So we're posterior of the urinary bladder here. Here the urethra coming down, right? Remember the urethra attach on the posterior side of the urinary bladder. The seminal vesicles are right here. All right, we'll get to that in a minute. These are the ampulla of the ductus deferens. So the sperm leave the epididymis, travel up through the ductus deferens, come down through the ampulla, and are going to be emptied into the ejaculatory duct, which is right in this region here. Okay, so that loop is associated with the ureters. The large end, the ampulla, is a region where the sperm or the spermatozoa are going to interact with fluid made from the seminal vesicles. Okay, so sperm are going to be moved through peristalsis. There's a ciliated epithelium here that helps move the sperm along to the ejaculatory duct, and then the sperm can be stored here as well for a while. So the ejaculatory duct is where you're going to find the junction of the ampulla of the seminal vesicles. Seminal vesicle with the ampulla of the ductus deferens. It's a really short region. It penetrates the prostate gland and it empties out into the urethra. And we remember the urethra from the urinary system. The urethra has two functions: the release of urine from the urinary bladder, and also for fertilization. So the sperm will move into the urethra with accessory fluids to transport the sperm for fertilization. And so what are these accessory glands? Remember, accessory glands are components that can be lived without, basically. However, without them, you will not have, at least in the male reproductive system, active semen. So the accessory glands are going to be making fluids that will produce semen, which activate the sperm for fertilization. So we have the epididymis and the seminiferous tubules. These only make about 5% of semen. It basically is making the sperm, right? The major functions of these glands, the accessory glands, the additional accessory glands, are to activate the spermatozoa, to give them energy to swim, all right? Give them nutrients or energy for motility, and also to produce buffers. It turns out that the vaginal compartment and the urethra themselves may contain a low pH or acidity, and these buffers bring the acidity or the pH levels to maybe not neutral, but it neutralizes the acidity of the urethra and the vaginal compartment so that the sperm can kind of handle the environment. The accessory glands include the seminal vesicles, the prostate, and the bulbourethral gland. So what do these things do? Seminal vesicles, which we saw in our last figure, are on the border of the bladder. Each vesicle has a branched series of tubules, and basically they're associated with the ductus deferens. And so what is a seminal vesicle making? A high fructose solution that helps activate the mitochondria and the sperm. So remember, mitochondria require sugars. High fructose, high glucose, basically a sugary solution that allows the sperm energy to move, motility. The prostate, what's that doing? It is, the prostate is a round organ. It's about 4 centimeters in size. It can get up to 8 centimeters later on in life. It encircles the prostatic urethra. So we call that, it encircles the urethra itself, but we call that region the prostatic urethra. And it has a bunch of tubulo-alveolar glands that are surrounded by smooth muscle. And so what's the prostate doing? It's making prostatic fluid. The prostatic fluid is a weakly acidic secretion. So again, it's a fluid that alters pH. And basically what this is doing is the prostatic solution is interacting with the female reproductive system to induce contractions to help ejaculate to move up into the female reproductive tract. So the secretions are ejaculated along with the sperm. The bulbourethral glands are a pair of little tiny round glands at the very base of the penis, and what these guys are doing is producing a compound that is going to be emptying into the urethra. And it's producing alkaline, sticky, thick mucus. And this also helps to neutralize urinary acids through the urethra, and it gives lubrication to the penis. And so the bulbourethral glands are producing another pH shift to help keep the spermatozoa healthy and happy as they're traveling along through the ejaculatory duct. So seminal fluid is going to include seminal secretions from the seminal vesicles, the prostate, the epididymis and sesenticular cells, the bulbourethral glands, and there's about 2 to 5 mils per each ejaculate that occurs, and that can include 20 to 100,000 million sperm per mil of semen. We'll continue with part 3 in just a couple seconds.